Welcome to the first podcast in a series of wrap-up podcasts that are designed to help guide you through this course material. So let's begin. Since the first GMO patent was issued in the US in 1980, the use of GMOs has indeed become a worldwide phenomenon. GMO use is a very hotly debated topic. There are strong arguments for, and there are even stronger, more voracious arguments against. So as we delve into this area, it's very important that we have an understanding of what GMOs are and how they're actually made. But before we can address how researchers have artificially created these novel combinations of genes that couldn't normally exist in nature, we need to have an understanding of nature's master code, the molecule that serves as the blueprint of life, DNA. This first unit deals with DNA. After this unit, you should have a full appreciation for the structure of this marvelous molecule. You should have keyed in on certain points, such as the fact that it's a double-stranded molecule, that the two strands run anti-parallel to each other, and also the fact that there's complementary base pairing between the two strands, which effectively zips this molecule up. DNA is tightly packed into the cells. Every living cell on Earth has DNA as its blueprint, and there's two basic types of cells. The prokaryotic cells, which have no nucleus, these are things such as the bacteria, and the eukaryotic cells, basically the rest of nature contains eukaryotic cells. They both have DNA packaged into them. This is a very tightly packaged structure, usually within the cell. So one of the main roles for DNA, you should have noted, is the information in it needs to be passed on to the next generation. Therefore, this code has to be replicated. So contained within this unit are the details of how this occurs for each cell. Just a fun fact to note here, since its discovery or description, the significance of our understanding cannot be understated. In fact, there's an entire day dedicated, DNA Day, dedicated to the discovery and scientific advancements, such as the very GMOs that we're studying, that this molecule, understanding how this molecule works, has made possible. FYI, it's celebrated April 25th each year, the same day that Watson and Crick published their transformational findings in their April issue of Nature. So how does this genetic code actually work? Because essentially the DNA is just the recipe, and the product is what you see as your living organism. So how does this DNA molecule become this? Life. Okay, the genetic code. How does it work? By the end of this unit, you should have gained an appreciation for the flow of genetic information, which begins with DNA and passes on to RNA, specifically mRNA, which is then used to synthesize proteins. So it is largely these proteins which perform most of the cellular functions and in fact generate the physical manifestation of the living organism, the phenotype. One of the key features of the genetic code is that it's universal, which means bacteria, the koala bear you see there, or even us, we all contain the same type of information, and it is used in exactly the same way that is presented here. So I pose a question to you. How has the universality of the genetic code enabled the production of GMOs? Ponder that. Okay, so we know that proteins are effectively the food that is created from the DNA recipe. What is the significance of proteins? Basically, proteins are the workers of this cell. They play a central role in every single process that goes on within a cell, basically. Each has its own unique shape, size, and function. There are millions of proteins contained within your cell. Okay? The shape of the protein is absolutely vital to how it functions. So, do you think altering the shape of the protein would change how it functions? Indeed, it will, in major ways. When constructing GMOs, when you insert a foreign gene, it's critical that the foreign gene be expressed but also even more critical, that the protein product is made and stably maintained within that foreign cell. Therefore, within this unit, we've given a discussion as to how proteins effectively take shape or fold into their unique shapes. So that ends the wrap-up. Summary for Unit 1. I hope this has helped, and I look forward to seeing you in the next wrap-up podcast.